Okay, ready for the final part, the panel debate. Thanks everyone for staying, also online. And the panelists are the speakers of today, Robin Waples, Thomas Kalnes, Victoria, Jane Reed, and Christina Lusso. Do this on purpose. And Anya and I have prepared some questions that we sent to the speakers beforehand, so they had some time to think about that, hopefully. And so this first part will be this questions that we're going to ask to each panel member. First about NE, 3 questions, then 3 questions about genetic indicators, and then we'll give the online audience or the audience in general 15 minutes for questions and answers, and then some closing statements again for the panel members. All right, so about effective population size. Today we have seen the theoretical construct, what it means, what are the underlying, what's underlying potential and underlying challenges, and then also how it can be translated into policy by translating them into genetic indicators. And during the whole day, there was a debate about precision, confidence, and so on. So there's a substantial gap between the complex concept of NE and the simple genetic indicators that need to be reported. So how much do we need to simplify when guiding stakeholders? Or how worried do we have to be? So that's the flip side. How worried do we have to be about genetic indicators being imprecise? I'll start with Robin.
I wish I'd reread the questions. So how worried should we be if they're imprecise? And what was the first part of the question?
Yes. Yeah, how much can we simplify, or should we simplify to make it workable? But then the flip side of that is how much imprecision can we tolerate?
Okay, so could you clarify, are you talking about actually simplifying the analyses, or are you talking about simplifying the explanation of what you're doing when you're talking to stakeholders? Because those are 2 very different things.
Yeah, in the end, stakeholders have to be able to use and apply and implement it and understand the concept. It has to stay workable.
Yeah, so I think some reasonable degree of simplification is necessary and not undesirable when communicating with stakeholders. And effective population size is really an elegant concept, but it's as soon as you start scratching below the surface, it can get really hideously complex. So I think it's possible to find an appropriate level to explain the concept to kind of regular people who are at least interested in trying to understand biological concepts. So I think that's definitely possible. And it probably will require some tailoring to, it wouldn't necessarily be the same exact approach to all audiences. Might have to maybe do a little research or thinking before talking to a particular audience, depending on their background or the sorts of things, analogies they might be receptive to. In the analyses themselves, there's probably going to also have to be some simplifications or compromises, probably, because a few data sets will exactly fit the models we have for estimating effective size. Monitoring genetic diversity, such as like monitoring changes in heterozygosity or so, that's a lot more robust and a lot relatively much simpler to do. Effective size has lots of nuances regarding both if you're trying to do a demographic estimate or if you're trying to do a genetic estimate. There are potential biases associated with all sorts of approaches. So I just have to probably use some reasonable approximations and so on when applying to actual data sets. That's a start. And my 2 cents.
Thomas?
Yeah. All right, thanks. Yeah, how should we simplify? I guess the talks that we heard today about the policy and these things basically that so this is a very extensive reporting that is going to be done and well, having spent months and months of doing the reindeer estimate for effective population size, you realize that that can't be done for every population in that way if you're going to reach over all of them within the short deadlines that is made. So certainly simplifications has to be made. But I think I'm sort of a bit of a pessimist when it comes to these simplifications, and I'm worried that the simplifications will take some of the usefulness of the estimates away by just generalizing this. Say, do you use some sort of rule of thumb for N over N, and then you have a census population size, and you get some numbers, but then at the same time, they're not very responsive to any dynamic changes if you have a harvested population or dynamic changes if you have populations that are really going down, for instance, and being reduced. So simplified can be okay, but I think it's very important that it is reported, these simplifications that is done. So we can really track whether your estimates are based on a 3 months work of getting an effective population size for this population, or whether it's just some rule of thumb that is applied, which is more or less accurate. So tracking these simplifications, I think, is very, very important.
Thank you. I'm going to start by also saying that if I leave in the middle of the panel debate, it's because I have to catch my flight, just so you know, now that we're running a little bit late on schedule. I think the previous speakers capture a lot of this already, but my feeling is that it depends a lot also what we want to use the information for. It's one thing when we want to monitor large-scale trends or this reporting to the CBD, but when it comes to when this is going to lead to conservation action, for example, I think we need to worry more about the imperfections of these indicators, more so than when we're looking at large trends. We've seen all the pitfalls, sort of the discussions during today, and all the considerations going into these estimations. And we know they're not perfect, but when it comes to policy and management, it's also important that they are pragmatic in a way. So sadly, we have to accept also that there is imperfections, and we have to simplify things in order to be able to get them into policy and management, in order to make it possible for countries to do these, calculate these indicators. So in that sense, I think that the proposed indicators and what has gone into the Montreal framework is a good compromise. And I think that it's good that we get it started, but surely the indicators can be refined as new knowledge comes about on how to make them more precise.
Thanks. Jane?
Yeah, I agree with the comments so far. I think maybe 2 things I would emphasize coming from the point of view of a scientist is I guess it's our job to be as appropriately sophisticated and as precise as we can manage, and that's kind of our job, right? Like Thomas says, is that those of us who are charged with trying to estimate these things kind of properly have to think about it and be careful about it and develop the methods as necessary and collect the data as necessary and make sure that process is transparent so that we we're always pushing the limits of how well we can estimate this number, whatever it is. I think then the communication can be extremely simple because it's more a concept than number. And then I think in terms of whether the imprecision or these kind of things matters, then it's definitely a case of what's going to be done with the number in the end. Because my experience with the Chuff study is that, we did as good a job as we can of getting a number, and it came out very small, and we estimated some kind of confidence interval around it. And, so we think it's in the right ballpark. But actually the number itself was very important in flagging up the need for conservation attention and getting government attention, getting funding. But nobody's actually used the number explicitly, right? Because then we went on to build a completely different population model that put in the ecology as well. And while we kind of checked that the output was kind of matching our estimate of any that we'd estimated from the real data, the number itself was never actually used in that prediction. And so in some ways, the number was a flag. It wasn't a quantitative entity that itself fundamentally underpinned policy in any quantitative sense. And my guess is that will be quite a common situation. So in that sense, I think pragmatism is the right way forward. And as long as it's clear kind of where you think the number is in the critical, it's fine, forget it, it's useless, then that's kind of the level that the government will be interested in. And maybe as an analogy, it might be interesting to think briefly about the kind of confidences that attach to other indicators. So, for example, in the UK, I'm quite heavily involved in developing indicators for the postulated impact of offshore wind farm developments on seabird populations, where, of course, we don't know, and there's a huge amount of uncertainty there, and a similar degree of complexity in what goes into the model to generate the magic number. And actually, I think what the government finds incredibly helpful there is if you can just attach a categorical confidence, like high confidence, medium confidence, no confidence. And that's actually what they use. They say, is it high risk, medium risk, no risk? Is it high confidence, medium confidence, low confidence? And that's actually what they're using. So I think in some ways it's our job to put things in those boxes as best we can, and then they can make policy based on that, but they probably won't use the actual number anyway.
Okay, thank you. Christina?
I like the traffic light analogy there. I can see that work definitely for policy and management. So I have two things. So the indicators that are currently in the Kunming-Montreal Global Biodiversity Framework got in, but there's also a call for additional indicators. So headlines, complementary component indicators as well. There are aspects of that Global Biodiversity Framework where for some of the targets there are no indicators. And there's even a pledge for refining and adding indicators also for genetic diversity. So there is an invitation also from the CBD so that they do welcome new indicators as well. So there is an opportunity to try to get more and refined indicators into the Kunming-Montreal Global Biodiversity Framework. So just wanted to mention that it's not as if what we have now is what we will have to live with for the next 30 years, not at all. And the second thing was, you also briefly touched upon it, Victoria. So it's, yes, we have these indicators, but they shouldn't just be a number in a country report. They should be used and used broadly by countries. And that is also the hope that there will be an uptake in these countries. I would say as it is now, many countries do not really have a spotlight on genetic diversity when they do biodiversity conservation. So just having that kind of mind shift in terms of thinking about biodiversity, where genetic diversity is part of that, is huge. And being able to actually use an indicator such as the NE500 would be a game changer. And yes, there is still work to be done. And yes, there is, of course, with the scientific hat on, we like things to be precise, etc. But we also, the pragmatism here is really key. So we in order to get people to recognize and actually use genetic diversity for conservation efforts is far outweighs kind of, at least to me, outweighs my need for things to be exact and precise. I would rather see genetic diversity really being adopted and used broadly for conservation.
Thank you. Since you mentioned, Christina, that more work is needed, I have a second question for the panel, and that's what are the main questions to be addressed in future research to fine-tune, optimize, get better grip on all these things, scientific questions for young scientists? Or maybe start with Victoria because she has to leave.
I think a lot of those questions have already been highlighted today, like these questions that has to do with how does how is it affected by migration rates or life history traits and so on. And there's a lot of work going on there. And when it comes to these different NE-NC ratios. But I think most importantly, I think it's about these interactions that Jane had so elegantly put in your talk about how these interactions between ecology and genetics and how these interplay in conservation action. And I think that's where a lot of work has to be done in what does actually these numbers tell us about what conservation actions are needed. How can these indicators be used and put into use when it comes to practical conservation action? And how does that interact with other type of threats that species are facing?
Yeah, sitting here thinking, where should I start with what should be the main focus? But I think in terms of really making these things applicable, we have lots of different types of indicators and making some progress on aligning how they are related to each other and how, say, genetic NEs are related to demographic NEs, and that we are sure that we are talking about the same quantity when reporting numbers. And this is especially for long-lived animals, where any estimation is far from easy, and it's a bit unclear really how you should do it if you're going to use genetic estimators, for instance, which are popular because it's well, let's say it's easy or less effort than collecting demographic data for years and years and then coming up with a number in 5 or 10 years time. So there's several things, but really aligning sort of any indicators and making sure that we're estimating the correct thing if you're going to estimate it. That is an important focus.
Thank you. Robin?
Yeah, I would, I guess, flag two things. One is I mentioned this morning the importance of getting a better handle on metapopulation NE because it's so important clearly for the NE 500 criterion. So that's really a complicated area, really need of a good relook at. The other was related to, I think the last slide I didn't show, I skipped a couple slides at the end that I didn't show. This one had to do with suggesting that people pay more attention to the possibilities offered by the fact that there are, you know, the three widespread genetic methods are often used on the same data sets, particularly the two single sample methods. It's quite common for people to report both estimates for the SIB-chip method and the LD method for the same samples, for example. And those are both estimating inbreeding effective size in the same time period. So in theory, they're both estimating the same parameter. So it should be the case that a more precise estimate can be obtained by combining the two estimates. And the appropriate way to do it would be to take a weighted harmonic mean of the two estimates, with the weights being inversely proportional to the variances. And I've looked, in fact, I have a manuscript suggesting better ways to do this. And that's nearly ready. The temporal and LD methods have a fairly simple way of getting theoretical variances as a function of true NE, the sample size of individuals and sample size of number of loci. So there's a pretty good basis for being able to properly weight the estimates. The SIBSHIP method does not have any analogous simple formula like that to generate the appropriate variance. But Wang has done a lot of simulations where he has published for selected scenarios variances associated with his estimator. So it could be possible to work his method into that as well. But I think there's a lot of potential there for increasing precision, particularly in there. And the increases in precision are going to be greatest where they're needed the most, where precision right now is the worst. So that's the good news. The other issue to consider is that the benefits of combining estimates will be greatest when they're uncorrelated, when they provide independent information about effective size. And I've done quite a bit of modeling indicates that the temporal and LD methods are basically uncorrelated. That's not true of the LD method and the SIBSHIP method. When they have small data sets, there's a lot of noise, they pretty much are uncorrelated. So for those situations, it would be considerable improvement to be able to combine those estimates. But when they both have, say, lots of loci, say 1,000 or more SNPs, they're both zeroing in on the same answer. That depends more on the vagaries of your sample size of individuals than it does other things. So you're getting more or less the same answer twice. So the benefits are not as great because they're positively correlated to a reasonable degree. But anyway, that's my second point. I think there's quite a bit of potential for increased precision for pursuing some combinations of methods, since people are already doing this, but rarely do they try to combine them into single estimates.
All right, thanks. Jane?
Yeah. Again, I think I largely agree with what's been said already. Clearly, the metapopulation / gene flow questions, which are not quite the same thing, are very, very important. And I think maybe then it comes back to what you also raised about the need to put these questions about NE in the context of the wider ecological and demographic constraint, which then kind of brings in different possible effects of gene flow, which of course is not just acting to increase the effective population size, but could have very different implications, for example, for the probability of evolutionary rescue or meltdown and these kind of things, depending on whether the immigrants are having positive or negative effects on the recipient population. So then it's not even just a case of knowing what the kind of metapopulation NE is, it's to do with understanding the degree of local adaptation and those kinds of things, which ultimately could cause very profoundly different impacts of any effort, for example, to increase NE by increasing connectivity, for example, where things could go very badly wrong if actually you're just connecting populations that will be better left apart. So I think ultimately to generate the correct management response as opposed to an indicator is to have that kind of holistic understanding of how the system is working, which is always going to be the big challenge.
Thank you, Jane. Christina?
Yeah, I couldn't agree more. But also, I think, so conservation is, I'm sad to say, all about people. So better understanding of the people aspect. So in Genemo, for instance, we have social scientists, and we have that for a purpose. That's because we work with different stakeholders. So a better understanding also is needed in terms of the various stakeholders. It could be landowners, it could be nature managers, it could be, practitioners in some form or way, and what is kind of hindering them from actually applying genetic diversity to species management, population management, area management, etc. So I think it's important also because we natural sciences, but I think we also need to look at the social science perspective in all of this, especially when we want to use the genetic diversity indicators more broadly.
Thank you. To continue with genetic indicators, so now we are rolling out the genetic indicators over the globe to start monitoring and reporting them. So my question to the panel is, do you see the inclusion of genetic indicators in policy as a game changer? I'll start with Robin.
You starting with me? Well, I couldn't hear the last part you said.
Do you see genetic indicators as a game changer in policy?
Yeah, I'm not sure. I guess it'd be nice to think so. There's an awful lot of people who spend a lot of time trying to get people's attention. I'm thinking back to some conversations I've had over the last year or so with Joachim Merge, who many of you know, I think. He probably would have been here, but I know he had some field work I think he had to get to right away. But I remember what he said one time is that it took us a long time to get people's attention, to really pay serious attention to the importance of conserving genetic diversity. And now finally we have their attention. So given that, we better deliver. That was the point he made. And so I got to say, that's one of the reasons I've been thinking about a lot of these issues in the last year or two and trying to finish some of the publications I've been just kind of thinking about that have been puttering around in my mind as a possible future product. That maybe this is the time to do them because the time is right and it's important. And if we don't deliver now, we may not get the chance later. So it's not really an exact answer to your question, but it's my sense it is pretty important, and we do have a chance now to make a pretty important impact. And a lot of people have worked really hard to get us to this point. So it's worth focusing now to try to pay so we can actually reap the dividends of all that hard work.
Okay, thank you. Thomas?
Yeah, and I would say also it depends. The good start now, certainly, and I guess sort of it took 30 years to get to a point that we actually now say that, okay, we'll actually start collecting data on genetic indicators, and that's certainly a huge step ahead. But then it depends on, okay, say you collect all this estimate and you need some plan for following up. So I talked with Stine, who's working at Artsdatabanken, and is doing the NE collection in Norway, and just asked, well, do you know something about the plans of Miljødirektoratet for following up these things? I said, well, I'm not sure they have any plans yet. So you're sort of lacking the next step, and that's sort of the critical step for whether this is a game changer or not. Whether we actually do follow up on these things and take it seriously and say that, okay, well, actually we have a problem here. We have a risk of losing genetic diversity. We need to make a plan to follow up with. Yeah, I think that's pretty much it.
Claudia?
I would say that getting genetic diversity explicitly into the CBD framework in this way is definitely a milestone, looking at how much work has been put into this. And, you know, obviously I work for Linda Leike, who has worked on this issue for a very long time. So I definitely view it as a game changer in the way that it gets genetic diversity on people's radar in a different way. We now, you know, countries actively have to report on this, which is a huge step forward and actually can set the wheels in motion. When I talk to people at Databanken in Sweden, and we talked about the possibility of doing this work together with the Red List, and they say there's no available data, et cetera, et cetera, et cetera. But at the same time, before, when the Habitats Directive came, they didn't necessarily have the data to report for the Habitats Directive. But when somebody says, here, you have to report on this, you find methods to collect that data, and it sets the wheels in motion to create sort of the infrastructure for that reporting. So I think hopefully that's what we will see with this as well. And I think definitely it's a milestone getting this work started. And I can mention as well that when we had our GNMO workshop in Sweden, we had one of the members of parliament there, and who was very excited about this and excited about moving this work forward and asking questions and so on to the government. And so now genetic diversity is being discussed during debates in the Swedish parliament. And I think that's definitely a game changer because it has not been on people's radar in that way before.
Okay.
I think for me, it's fundamentally not a game changer in that the game is just the same, in that we want to try to conserve biodiversity in the face of many other largely anthropogenic pressures that include agriculture, fisheries, whatever else. So that game is exactly the same as it was. And that's the fundamental challenge. So I would see it more on the lines of having an extra player on the field on our team, because in order to push back against those other challenges, the land and marine environment use, the government needs to demonstrate some criteria that it has to act on in order to fight back against the economic interest. And I think, you know, the ones that at least in Scotland currently apply include things like, you know, if you've got a population that's on the kind of Annex 1 threatened list, whether your area contains more than 1% of the adults, or whether, you know, contains more of the breeding population, these kind of things. But if there's another criteria that comes along that says, okay, you need to maintain your population size above this kind of threshold, then I can see that can be quite a powerful tool actually in order to help the government officials and conservation agencies who are already trying to achieve these things to have the kind of evidence and legal weaponry that they need in order to try to achieve some of these things on the ground. So I think for me, that's in a more practical sense, that's going to be the major contribution rather than actually a fundamental change in the game of what everyone's trying to do.
Yeah.
So yeah, I don't know. I think most of it had been said before. So just reiterating, you know, if things are not in policy legislation, countries are not going to do much about it. So in that sense, it is a milestone. There is definitely also room for improvements. Just if you look, for instance, at the EU, there's a new EU nature restoration regulation, a complete absence of genetic diversity. So just to say that there is much more to be done, both regionally, internationally, on a national scale as well, to get genetic diversity into legislation and policy, because that's the only way that, you know, things are being done. So the different ministries and agencies responsible will not do anything unless there is a mandate for them to do that. And that mandates come from policy and legislation. But yes, it is a milestone, but there is so much more to be done. Fortunately, there's a lot of great efforts ongoing already, but we need more. So Europe has many initiatives ongoing, but globally there is still a lot to do, and especially in biodiversity-rich countries and regions. So yeah, we still have a lot of work to do, as Joachim said.
Okay, thank you. Then it's time for questions and answers from the audience, both in person and online. And we start with Chaton.
Thank you. Yeah, yeah. I have a question. I have a very skeptical mentor, like not him, but another one. Every time when I mention genetics, he will say, you can also do this phenotype. So I was just thinking about, because like we go very directly from with genetic data to a proxy without considering anything about the phenotypic variance. If we consider a trace and the heritability H squared, it's the same. And then the question becomes a VP over VP plus V environment. And this is the same. And then if we use a ratio of VP over V environment to observe the change of this ratio, would that be enough to observe whether we have genetic change? Because if we have a higher drift, that means this ratio will increase. And we have lots of data of phenotyping.
Who's the question?
Anyone? Everyone? I don't know.
I would say that in some cases that kind of thing can work. If there's a particular continuously distributed phenotype they're interested in, then there is an argument to say that you can estimate the G matrix more efficiently from the P matrix than by estimating the G matrix, given the amount of uncertainty that's often included in the latter. I would say, from my point of view, if the ambition is to predict population growth rate, when then the fundamental traits are the demographic rates, then that approach will not work, because most of the genetic variation is latent, and the degree to which it's actually manifested changes with the phenotypic mean. And so the whole idea that the heritability is just VA over VP doesn't work anyway. And so at that point, the kind of translation to just estimating heritability from the phenotypic variance over the rest certainly won't work. So I think for me, it depends whether you're trying to protect genetic variation in a particular trait like body size or wing length or something like that, versus whether you're trying to protect adaptive genetic variation that would actually allow rapid evolution of population growth rates.
Any questions online? Yeah.
Yes, should I read them out?
Sure.
Okay. Yeah, we have several. You have to stop me then. So the first one is the policy focus is so often to maintain NE or to maintain N to prevent population collapse. But in what case might we want to actually increase NE, and how do we achieve that?
Anyone? Jane?
Oh, it's a good job you gave me a microphone, isn't it? Well, I would say, for me, part of the answer to that is what we've already said, is that trying to maintain N is not very different from trying to increase N. It's only a small difference. And I think then, in terms of how to increase NE conditional on N, there are some other routes, which is what Henrik was just touching on at the end, whereby the kind of demographic decompositions that he and Thomas and I did in the trough actually tell you which vital rate and life stage is the one that's causing the demographic variance, which is typically, I think, to do with prime age adults and their reproductive success. Maybe Thomas would have a slightly different view, but that in many systems. So then at least it would tell you where to focus an intervention whereby you could increase the NE to N ratio. So that is another way in that could help things a little bit.
Victoria?
I think that part of the ambition with these genetic indicators is to also highlight where action needs to be taken. So if you have populations that are below this threshold, it is an indication that action should be taken either by trying to increase population size and thereby increasing the NE, or by increasing connectivity. I think those are the two main options.
Thank you. Any other question?
I could just add one thing. There is quite a bit of track record of people in captive propagation using various methods to reduce variance in offspring number that have been more or less successful. Depending on the life history of the species, it may be with salmon hatchery or fish hatcheries. For example, it may be fairly easy to equalize offspring number at the egg stage. But when you release the juveniles, that's your last chance to equalize family size. It may be that population experiences really high mortality after release. So it can only be partially successful in reducing family size variation. And it's, to do this in wild populations would require some active meddling, which might actually make things worse rather than better. So in the wild populations, I'd agree with Jane, the best option is just try to increase N and let N follow.
Just following up on that as well. So my background comes from the zoo world. And what we do is small population management. We do also play a role in terms of if we have wild populations that are not thriving and there is a need for ex situ breeding, then we do also play a role in that. And that is also one additional way to help boost population sizes. There was mentioned previously in terms of seed banks. There are also various biobanks out there. There are also, these gene banks where there are samples, there are cell lines, etc., that can be used to kind of recreate individuals, adding lost lines of populations as well. So there are ways also to kind of help boost populations in the wild by means of ex situ or gene banks.
Yeah, since you're all talking, I might just well add something here which came to my mind, because it's like you're already doing a lot of small population sizes with red list and already sort of categorizing species, trying to identify species where there's concern and we have to do something. And of course, any part will be adding to that, and we can identify species. But then it also struck my mind, well, okay, so we are really country-based again, same with the red lists, and all of these things. But then lots of the efforts we need to do might actually be cross-boundary, and we may have cross-boundary populations, or for instance, the white-fronted or less white-fronted goose. We have them in Norway for 3 months, and then they go to Greece and they stay there for the rest of the year, and then they come back to us. And in the between, they have to visit all these other countries. We might have the NE estimate for the breeding population. Greece might have an NE for the winter population. And there's a lot of collaborations needing to be done here, which requires international efforts. And I think that's an important part here.
This is a very good question because everyone keeps answering.
Yes, and there's actually an EU Biodiversity Plus pilot project ongoing with transboundary species, which Joachim is co-leading on as well, because definitely there is a need to look into that aspect, and it will include several countries and focus on different taxonomical groups, but that's something we can take later. But yeah, it's definitely something that should be investigated further.
And I think another important point here is when we're talking about what action needs to be taken or what is possible, to consider why is the population so small? Why do we have these issues? And I mean, it's possible to do translocations and so on. But as highlighted earlier, there might be other factors that one needs to consider in the sense of habitat quality and so on. So even if we do efforts to increase breeding and so on, it might be possible that there are other factors that we need to take action against if we are going to be able to have a viable population in that area.
Okay, thank you. Anja, since the online people still outnumber us, were there other questions?
Yes. So one question is somebody's asking if you could talk a bit more about what to do with like populations of species that really clearly don't fit, like a priori a right fisher population model, like clonally reproducing systems.
Okay, what to do with the hard cases? No, nobody jumps to answer this question, Anja.
Okay, that might be disappointing for the person who asked.
Well, Thomas going to give it a try.
I can say something at least. Something I'm thinking a bit about when it comes to really like applying rules of thumbs of these approaches. Whether you should, instead of applying one rule of thumb, try to develop some sort of rule of thumbs. How sort of categorize species and by sort of the different properties of the species, how long-lived they are, how they reproduce, overlapping generations or not, might come up with some better sort of rules of thumb. So if you're going to use a rule of thumb, you might instead use this rule of thumb. So yeah, I don't know. That's some sort of a answer, but it's difficult. And the best, of course, would be to actually do an estimate. And then if you don't have one for that species, find a similar species.
Yeah, I have just a quick comment on that. In this genetic indicators and 9-country study work in the guidelines, we have tried to describe different types of cases and how to think about different taxonomic groups or different species with life history and so. So we have tried to include some guidelines on that. However, there is still more work to be done, and there are several ongoing initiatives to produce even better guidelines for how to deal with different types of species and more challenging groups.
Thank you. We can take one other question in the audience. Yvan?
Thanks. Yeah, okay, so I see the need of like defining those indicators in terms of like policy making, and I think like Jane gave a really super convincing example of when you have a small population, you need to communicate that it's doing bad. I was wondering if we should be worried about the fact that by creating simplified indicators, we maybe give away a little bit the power to say it's complicated and to say we don't know. Because, for example, if this NE500 becomes like widely used, then do we lose the power to say, yes, this population has NE that is super high, but that doesn't mean that it's not endangered. That it doesn't mean that we have a free pass to just destroy it because it has a high NE. So should we be concerned about that?
Jane?
I think we should use all of these things carefully and make sure that they're used in context, and that's true of all of these kind of governmental indicators, of which there's a bunch, right? Because, you know, for example, in Scotland, there's a whole bunch of different indicators that are already in play. Ones that are to do with the new Scottish biodiversity strategy, which are to do with kind of basically, not in a very vague sense, not letting things go extinct, but it's not nearly as kind of specific as an NE one, which I think will then feed into that. So I could see the way that this is not being taken in isolation, but being taken alongside other indicators, where then, as I already mentioned, there's a renewable energy indicator impact renewable energy impact indicator that's also feeding into that kind of process. And there are probably other ones that I'm not party to that. So I think the people that are using these things are already kind of aware that it's kind of a multiple information flow feeding into the decision. Maybe that's the best way to put it.
Yeah. Second that. So putting things into context and never see things as geneticists, we also sometimes look at different populations and whether, taxonomically we should consider them as separate subspecies or even species, etc. But never taking the genetic results as kind of the only means to which to kind of define whether we should elevate something into a species, for instance. There should be other factors taken into account. But in terms of this, I think one of the examples, and I think you showed it, Victoria, in terms of the red lists. So the IUCN red list is used globally, and it's so well known. Everyone knows the IUCN red list almost, but it's not really reflecting the categories, doesn't really reflect genetic diversity. And I think you showed that figure as well. So also, yes, use the red list, but with caution. And now we know also that there are pitfalls in terms of genetic diversity, for instance. Likewise, with the NE larger than 500, should we just say, oh, everything's fine and dandy? No, we shouldn't. Of course, there's the context to everything. And if we see that there's extensive habitat destruction, etc., it could be a population or species going drastically towards decline anytime soon. So putting things into context, I think, is super important here.
I agree. I think it's really important to have this sort of holistic more view of looking at a species as a whole and what are the different threats and so on, and looking at different aspects and not just considering this one parameter. We also have examples of species that might be well below NE 500, but we still don't need to be specifically concerned because they have evolutionary strategies to cope with that. So I think that, this threshold is not necessarily a given, that it just means that we don't need to be concerned or that we should be concerned. And I also have another example from the red list. The red list is considered globally and so on, but it also has its imperfections. And one of the examples, the red list has these thresholds, right? And the moose in Sweden is red listed now because it's declining rapidly. But the moose is a very widespread species. We have a lot of individuals if you look nationally. So how concerned should we be? But it's red listed because if you look at this criteria, it fits into these threshold values. So it's one of those cases where maybe you have to put things into a context, really, even if you get a warning signal. So I think that's very important to consider. And I'm going to have to leave now. So I'm going to say before I leave that one of my hopes for the future is for genetic diversity and genetic vulnerability to become more integrated into the red list and the red list system, since it is so globally accepted as well. And I think that, right now the red list looks mostly at extinction risks, but I think it could be very beneficial if it could also take into account the risks of genetic erosion and what that means for long-term survival for species.
Thank you, and thanks, Victoria Koppe, for joining us. Anja, how many more questions do we have online? Is there one very important one?
If I say no now, I'm offending people. I have 1, 2, 3 more. You can pick how many you want.
Just do one more?
Yes, okay, but which one? Yeah, maybe this one. So, going beyond NE-based indicators and towards more comprehensive assessments, somebody's asking, if we think, beyond individual species and more about ecosystems, that in addition to what we already have with these indicators, we would also need to include some kind of risk assessment or exposure assessment of exposure to cumulative human pressures in the indicators somehow. And they want to know your opinion about this.
Any opinions? Thomas has an opinion.
Yeah, well, so there's different sort of scales and different things here. And when you talk about the genetic diversity, it's sort of on a scale of generations often that we are worried about the small effective population sizes. But when they're talking about habitat degradation, area transformation, these types of things, it's processes that goes much, much faster. And certainly it is important. It's very important. And there's loads and loads of focus on how fast we are now already transforming civil habitat for natural species into human feasible areas. And some sort of an indicator for habitat transformation is really critically needed also.
Yeah, and I guess in some ways it comes back to the idea of joining the dots and having some kind of integrated projection, which builds the genetic phase into the ecologically and environmentally driven parts of the demographic change. And I think in some ways it's kind of interesting that there's a long tradition of trying to do that in the context of small or decreasing population size and hence escalating inbreeding, right? That's been an ambition for a long time, and those models have been increasingly kind of combined with submodels that also factor in the driver of ecological change and hence the initial cause of population decline, these kind of things. I think to my mind, and I may just be naive or ignorant of some kind of coherent literature, but I think then that literature has historically been quite divorced from the kind of parallel literature that's then trying to look at adaptive genetic variation in the same context. So there's a whole kind of industry looking at evolutionary rescue at the same time as the industry that's looking at kind of inbreeding induced extinction vortexes. But I think for whatever reason, there hasn't been all of that much where everything's happening at the same time. And I think that's where actually NE becomes the key part, because it can help us link both of those parts. And I think there is some work that's starting to do that, but then the number of studies that's actually parameterized not just, a lot of empirical studies have either parameterized the inbreeding part or the adaptive genetic variation part, but not both. But now we are starting to see some empirical studies that have all of those parts and can put them together. And I think that's when we start to get this holistic ability to predict the outcome.
All right, thank you. Last question from the audience, and then we have a closing statements by the panel members. Or, just go ahead.
Yes, I just something came to my mind because we talk a lot about the simulation, the rice fisher model, and many species doesn't follow. I was just thinking we have enough, not enough, but we have actually really high power for computations. And those stepping stone model kind of model is kind of abstract of the real system, make it simplify, but we can go further to make even more complex. For example, like biophysical model in the marine system, they just give the behavior of individual and run the Lagrangian model and just trace the change over time and finally see how the change, the population structure, meta population, so on and so forth. I think, would you agree that what do we one effort is on the life history tree, because we actually don't know what exactly how life history tree changed the NE estimate and how the life history tree changed the meta population structures, so on and so forth. So maybe there could be something, I don't know, but what would you think that the most important life history trait that can actually affect our simulation? I don't know.
So your last question was, if you're going to model realistic life history variation to simulate genetic diversity or estimation of effective size or something, what would be the most important one? Yeah. Well, if it's related to effective population size, the two most important things are, one is generation length, because generation length, long generation length associated with large effective size. So traits associated with longevity, changes in fecundity with age. So if you're increasing fecundity with age, for example, that's common in many species, not mammals, for example, but marine ectotherms, for example, have indeterminate growth. They usually get bigger and more fecund. That increases both generation length, probably also increases variance in reproductive success. So those may trade off. Those are some of the most important traits. The other, besides generation length, I would say is lifetime variance and reproductive success. That's a huge determinant of NE to N ratio and traits that are associated with that. Some of these persistent differences, for example, that can really make a big difference. That's not a single trait, but that's some of the ones that I see as most important.
All right. Now, to finish the debate, I'm going to give the panel members a chance to make a last closing remark about what is their hope for the future, or where are we in 10 years in the field of conservation genetics. I'll start with Christina.
Thank you. Is there hope for the future? Yes, definitely. I think that there are so many good people working on this, and there is a mandate, there is will, there is potential. And there are so many good initiatives also rolling. So I think there is definitely hopes for the future in terms of actually using genetic diversity for biodiversity conservation. But there is a lot to be done yet. Still, sorry, definitely. So yeah, we will be busy in many years ahead.
Thank you, Jane.
Yeah, there's always hope, but we need to be quick, right? Because things are going down the tube, so we can't mess around. And I think we're increasingly starting to have the tools and the data sets and the people that are able to bring those things together to affect some very important changes. I think there's some traps for the scientific community that we need to be careful not to fall into. And this is not to do with anybody here, I should add. I mean, I think one obvious trap is the let's just sequence everything school of thought, which I think is going to be extremely insightful in some ways, but probably a massive diversion in other ways. And I think, again, what we need to do is make sure, as Robin alluded to earlier, that those kind of data, which can be tremendously empowering, are collected wisely, used wisely, in the context of other things that people are starting to forget about because it's easier just to sequence stuff. So I can see that as a risk that we need to be careful of. If there's too strong and simple a voice that understanding genetic variation is the key to everything, there's a very obvious misinterpretation of that, which is that we just sequence stuff and it's job done.
All right, thanks. Thomas?
Yeah, right, thanks. I think my hope for the future would be that we were able to not forget about demographic needs when we're actually thinking ahead and trying to really understand how we can estimate these parameters in a good way and how to relate estimates of these local populations to the more large scales, like the meta-population estimates, and really being able to use the estimates that we are collecting. So if these indicators are supposed to be useful and applied in sort of a bigger scale, it would be good if we could track changes in these indicators. And that is sort of, if you are going now about with just doing proxies and doing it rapidly, and later on we do it in a different way, we can no longer sort of see any change in these indicators and really interpret them in a meaningful way. So my hope would be that we are doing things in a way that it's traceable also, and we can really see later in the future, if we repeat it, we could be able to see whether have we improved actually or have we not, and at what level have we actually managed to do any improvements in these indicators.
Thank you, Robin.
Yeah, so I'd like to reiterate what the last two folks said, having to do with population demography. And it's fine to use genetic methods to estimate effective size. I've worked a lot on that myself, but to me, the gold standard for effective size is being able to reconstruct population pedigree. And there are a variety of ways of doing it, but some of the best involve molecular methods for pedigree reconstruction. And in general, you don't need a million loci or whole genome sequences to do that. You can probably do most of it with a good microsat data set. So with increasing attention in the future toward potentially regular monitoring long-term data sets, we have some real potential to build up long-term pedigree-based data sets for populations that can be enormously useful for all sorts of things, some of which involve rigorous estimation of effective size, but there are also all sorts of other things, heritability estimates, all sorts of things like that. So I think there's going to be increasing opportunities to do that in the future that we ought to try to take advantage of. Collectively, scientific community.
All right. With that, we come to the end of the day, of the 2 days for many of us. So thanks again for all participants and speakers of today and for the interesting panel debate. And the final word is for our host, Alex.
Yeah, I can only say thank you. I think the presenters did a stellar job on this very cumbersome, but also challenging, but also super interesting topic. I think everyone, at least if I speak for myself, I learned something. I got very stimulated by the presentations. Also the sometimes challenging questions. I think it's good maybe to challenge also the presenters. So it shows that where we are at a moment in this topic. So even though it was also a bit challenging to organize everything, but I think in the end it went very well. I mean, the number of registrations, especially online, speaks for itself. With over 170, 180 registrations just online. So I hope it was also bearable for you online. I know that there has been a bit of a problem with the sound in between, but I think we kind of fixed that. And also thank the audience and guests who came here very, very much. I think we had a good afternoon, good day with it. And thank you, everyone.
And one remark for the online questions, if you can keep track of the unanswered questions and distribute them around, and you might get an answer or not. Thank you.